Hello everyone, and welcome to our online lecture series for plant biology. Today we pick up kind of where we left off in our last lecture, because as I said last time, we're in the middle of a three-part series on movement of water, minerals, and nutrients throughout the plant. And so last time we covered in great detail how the structure of water leads to different principles of water. Namely, the fact that water has cohesion. It's able to pull itself through those slight positive and negative charges between the hydrogens and the oxygen in order for it to stick together. This is the reason why we have rain droplets, not rain atoms. This is the reason why water stays together when it gets spilled. And this is the reason why water forms a consistent column throughout the xylem of a plant, being pulled up through the process of transpiration as water leaves through the leaves, the stomata in the leaves specifically. Now, there are a couple other forces helping pull the water up the xylem in the plant. The water itself also has adhesion to the cell walls of the xylem because they are charged in such a way as to interact with the hydrogens and the oxygens in water. They enable the water to bind to it, as illustrated here in step 2 of our diagram. Now, the last thing that we talked about, and where we'll be picking up, or the last major vocab, and where we'll be picking up the baton and carrying it today, is the idea of diffusion. The movement of things from where they are in high concentration to where they are in lower concentration. Diffusion governed our source sink model of movement of sugars through the phloem, where there was a lot of sugar dissolved in the water of our leaf cells that then got transported to our companion cells where it was at high concentration. And then through a concentration gradient, it was passed down the phloem up until it reached its sink in the root cells where it was converted into starch, a non-dissolved sugar that did not mess with the ability of diffusion in order to drive the movement of water up and sugar down the plant. Now, the diffusion of water is specifically referred to as osmosis. And it is in a discussion of osmosis that we'll start our lecture today. So let's talk about what's happening at the cell surface when you have water moving in or out. So here's my attempt at a diagram for the cell. You've got the external cell wall made up of different kinds of fibers, like the green cellulose, providing the major backbone for the cell wall, the light green hemicellulose, and then the linkers, the pectin. Oops, not pectin, but pectin. And inside of the cell wall, you have the plasma membrane, made up of its phospholipid bilayer, so the fact that it's got a water-loving or hydrophilic outside, hydrophobic blue tails, so that way water, as illustrated by our little diagram here, can exist happily over here or inside the cell, but you will not find water within the two layers of the lipid bilayer. It has to be either on the outside or on the inside. It cannot ever pass into the cell via the plasma membrane. Now, we also know that or rather, sorry, vocab term here. The fact that the plasma membrane is able to control what is coming in and out means the plasma membrane is selectively permeable. It picks the things that are going to come in and out. And this matters a lot because the whole goal of today's lecture is to understand how water is filtered before it gets to the vascular column of the plant. How do those roots that are uptaking water, how do they do that filtration step? So keep that in mind as our goal for today. Now, as other components of the plasma membrane, beyond just the lipids that provide that hydrophilic, hydrophobic barrier, we have some transport proteins. Like this purple protein here. Transport proteins span the plasma membrane, meaning they go from the outside to the inside, and can specifically allow certain molecules to go through. In so doing, they manifest the fact that the plasma membrane is selectively permeable. Those proteins are what are picking what is coming in and out. This protein in particular is called aquaporin, or water hole, because aquaporin is the specific transport protein that allows water to come in. It adopts a very specific conformation, wedges, a little bit too small there, let's make it a little bit bigger. It adopts a specific conformation, binds in to the transport protein, the aquaporin, and then falls down in such a way so that way it matches the pathway exactly, and now it can get inside of the cell. If you were to have a sugar molecule, here's a glucose, that would come in, it would not have the right shape, and the transport protein aquaporin would not let it through. So aquaporin is a door specific for water. And water will be able to go in or out through these aquaporin transport proteins. It's constantly going to be able to move across the plasma membrane based on where it has its differential pressure through diffusion. So it'll be going to places where water is lower in concentration from places where it is higher in concentration. Now, as water is moving around, it's going to actually change the structure of the membrane itself. When the membrane is full of water, it actually gets pressed up against the cell wall. It moves closer to the cell wall and keeps the cell wall structure very rigid. This is what we call turgor, or turgor pressure. This is what happens when the plant or the plant cell has a lot of water, and therefore the plasma membrane pushes out the cell wall. Well, if the plant has turgor pressure when it's full of water, what do we call it when the plant lacks water? Well, a plant that lacks water is wilted. And wilt is driven by the fact that each and every single cell in that plant has not enough water, and therefore the plasma membrane is not pushing the cell wall out, and therefore the plant adopts a very specific look to it. Y'all will be familiar with a plant that has full turgor pressure versus a plant that is showing wilting. So let's check out a cool video of a flower wilting. Going to Wikipedia, this time-lapse video is quite cool, I think. You can see the flower start out here very rigid. It's actually able to overcome the force of gravity by sticking that flower straight up. That's how strong the water pressure is. But as that water pressure decreases, we start seeing the petals become softer. They're not standing upright against gravity. Instead, they're starting to fall down. And this is not because the skeleton in the plant is breaking. This is not because the cell wall is falling apart. This is simply because water is escaping the plasma membrane and no longer pressing against the cell wall. When it no longer presses against the cell wall, the flower wilts. Kind of cool, I think. And now that we're back inside the cell, or looking back at the cell, what's the organelle in the plant cell that's actually controlling how much water is inside of the cell, or being able to stash water for times of need? Well, that is our vacuole, our water storage bag. So we can look at the interplay between our cell wall that's allowing water to kind of move around it. Water's always trafficking around the cell wall, floating around because it has the correct charges. It's a polar molecule, so that way water can interact with it. Just think your paper towel and water. Paper towels are made of the same things that cell walls are, so water grabs a hold of our paper towel or cell wall. If water is in high concentration surrounding the cell wall and in lower concentration inside of the cell proper, well then that is going to drive water from the outside through our aquaporins inside of the cell. And when water is rushing inside of the cell, this causes turgor pressure. Well, when a cell is stored in an environment where there's tons of water, and the vacuole is being pushed in such a way so that the plasma membrane is being pushed against the cell wall, causing a lot of turgor pressure, we call that a hypotonic solution. If instead there is a shortage of water, causing the vacuole to shrink down, and then the plasma membrane to not push against the cell wall, and this is a cartoon of how much pulled in it can get, so this plant is incredibly thirsty for water. Water has left the cell because it's evaporating out and leaving. This plant would be wilted on the whole, and specifically these cells that have their plasma membrane collapsing in are going through a process called plasmolysis, or the breaking of the cell due to a shortage of water because the plasma membrane is collapsing. This type of solution, this type of environment, is hypertonic. Now, if there is a balance where water can move in and out at the same rate, the plasma membrane may not be pushing super strong against the cell wall, but it's not totally collapsing. That kind of solution would be considered isotonic. Now, the plant usually wants to exist in a hypotonic solution. It wants to make sure that the things surrounding its cell wall are literally just water. This means that there will always be a large concentration of water on the outside, a lower concentration of water on the inside, therefore water will always be rushing in. So the purer the water that exists floating around the cell wall, the happier the plant cells will be. Let me go ahead and say that again because it's going to be crucial for our entire lecture. The purer the water around the cell wall, the happier the plant cells will be because water will always be driving into the cell, causing turgor pressure. And with turgor pressure, the plant is standing upright, so that way it's absorbing the most sunlight and performing the most photosynthesis. It's the most metabolically active. It is growing the most that it can. And so this is how these different types of solutions, hypertonic, where there is more water inside than outside, so water leaves. Isotonic, where there's a balance of water, equal parts inside as outside, so water moves in and out at the same rate. And hypotonic, more water outside than inside, so water will constantly be rushing in. Matter in terms of our ability to move water throughout the plant. Now, I will say that in a hypotonic solution in turgor pressure, the plant cell isn't going to explode. If you were to do this to an animal cell, you'd actually explode animal cells. They don't have an outer barrier. The cell wall itself provides a physical restraint against the plant cell getting too large from too much water. So it's not that it's going to die because it pops, like you or I would if we were to put a garden hose down our throat and turn it on full blast. You have no upper limit, so you would literally rupture, just like your body would, your cells would. Plant cells have that plasma membrane, have that cell wall that restricts the expansion of the plasma membrane. And so in order to understand how a plant is filtering water in order to try to get the purest water surrounding our cell walls and plasma membranes, we have to discuss uptake of water in the roots. And of course, before we get there, we have to talk about 3 different ways to move water throughout the plant. When water moves throughout the plant, it can move by contacting only the cell wall. Looking back at our diagram, this means that these water molecules, let's see if I can grab just them. Cool. These water molecules will flow around the cell wall of one cell, find the cell wall of another, find the cell wall of another, and constantly flow through only cell walls. They never pass into a cell itself. This type of movement is called apoplastic water movement, and it is only on cell walls. If instead water moves from cell to cell via the cytoplasm and specifically transports to the next door neighbor cell via a plasmodesma or plasmodesmata, that means that it has engaged with the aquaporins. That means it's been filtered. And that's the major difference between our apoplastic movement, only cell walls, so this water is unfiltered in its movement, and symplastic transport, meaning that it moves through plasmodesmata. By moving through plasmodesmata, it at some point had to go through an aquaporin. And by going through an aquaporin, you ensure that it's just the water molecule and not anything else that's inside of the cell. So moving through our plasma membranes means that you get filtered. Oops. Are you seeing where we're going with this? There is one other type of movement in addition to our apoplastic and symplastic pathways called a transcellular pathway. And this is where the vacuole gets involved. So just like the plasma membrane has aquaporins, the vacuole also has aquaporins. So this basically means you're getting filtered and filtered again. Not a lot of water moves transcellularly in a plant because it just requires such a strong bottleneck of going through only aquaporins. If we're looking at how much water each of these three different modes of movement are responsible for moving, well, apoplastic is the most because it doesn't have to wait for an aquaporin to be open. Symplastic is second most because it only has to go through one set of aquaporins, and transcellular is the slowest, third most. Not a lot of water goes through the plant transcellularly, but it does offer another way to move water through when you need a lot of water really quick. And so this illustration from Wikipedia, again, illustrates elegantly how water can move through the apoplastic, just hanging out on the cell wall. It's got adhesion to the cell wall, so it's going to keep funneling through. And as long as there is more water over here, as long as there's more water over here than there is over here, water will continue to traffic along the cell wall, just like a paper towel absorbs water in a puddle, right? The water will keep absorbing into the paper towel into regions where water is not yet. In our symplastic pathway, you've had to have the water from the apoplast move into the cytoplasm and therefore be filtered via an aquaporin, and as it moves through the plasmodesmata, there are aquaporins there as well, and so the water is filtered. So now how does a plant decide whether their water will be moved apoplastically or symplastically? Well, there isn't actually much of a decision with this. It's just which road is available. Think of cars driving on a freeway. If the freeway is open, freeway being apoplastic transport, lots of cars will jump on it and move very quickly throughout the plant. But if the freeway is starting to get clogged up and there's a lot of water moving there and therefore traffic is moving slower, they might see some side roads open, symplastic pathways, and hop over there and move over there. But there isn't a reason other than availability of the roadways, unless the plant forces it into the symplastic pathway. And that is the crux of water filtration. No, I guess we can write in blue here. Water filtration in the roots is all about forcing all H2O, all water, to go symplastic. Even though it may have moved initially through the roots via apoplastic absorption, it needs to be forced symplastic in order for the water to be filtered. So we want to find a reason in order to push it into aquaporins and keep it out of the cell walls. Well, let's take a look at the structure of the plant root that allows that to happen. So here is a model of our plant root. Y'all will recognize the different tissue layers. We first have our outside of the plant root over here, which has just been freshly watered. So these arrows indicate that water is rushing in. Water will first rush into cells of the epidermis, which of course have their own cell wall and cell membranes. So cell walls are indicated by the darker color and cell membranes begin where the lighter peach is. Now, from our epidermis, we have cells behind them that are of the cortex. Behind our cortex, we know we have the endodermis, a skin layer inside of the plant. And then behind the endodermis, we have our pericycle, and our pericycle is our last step before we reach our xylem, our vascular column, or our stele. So you'll hear this referred to as our stele. That is the shape of our vascular column. Now, when water is first entering in the epidermis, you can see that it will be transported apoplastically when possible. Water is moving through the cell walls as the roads are available, but then there's a structure in the endodermis, a very particular thing called a Casparian strip that is now going to provide a roadblock. No more apoplastic transport will be possible. Instead, all of the water is now transported symplastically via those plasmodesmata. And so it's something about the Casparian strip that is forcing water to go through the plasma membrane and no longer be able to be trafficked in the cell wall. So I'll give you all a moment to kind of absorb this diagram before we discuss our details of what exactly about the Casparian strip allows it to really force water in the way that it is doing so. To uncover our last two blanks, we see apoplastic movement either through the clear spaces of the xylem or the dark spaces of the cell wall, and all symplastic water movement through the lighter peach-colored spaces in each of our cells. So, what is it about the Casparian strip that forces water from its apoplastic movement to symplastic movement? I mean, didn't we talk all about the fact that cell walls are built out of cellulose, hemicellulose, and pectin, which all do a great job of conducting water because they can bind to water and reinforce that movement via adhesion? Aren't cell walls designed to do that? Well, yeah, for the most part, except for when they're not. And the Casparian strip is one of those examples of a region of the cell wall, a particular layer of the cell wall that's designed to prevent that movement. So let's take a look at two images from your textbook about how the cell wall is built and those three different methods of movement. So first and foremost, here we see colorings of three different layers. You see the actual background of the cell in gray. So you have like the cell wall structure in gray. And then you've got a strip, the Casparian strip, in yellow. And it is layered in the cell wall to prevent any of the apoplastic water movement across a very specific layer of the endodermis. So this is really what defines our endodermis, cells that have a touching Casparian strip. Cellulose is still a large part of the cell wall, even in the Casparian strip, but we've got the addition of two major other molecules that help define the Casparian strip. The first is suberin, and the second is lignin. And we'll take a look at these in a second, but looking at our other image from the textbook, you can see that whether water starts out apoplastically, traveling via cell wall to cell wall to cell wall, cell to cell, when it reaches the endodermis, The Casparian strips prevent apoplastic movement, and therefore that water has to go from the cell walls into the cells and then can go back to apoplastic movement till it reaches your xylem. And this is where your xylem's at. Now, if water starts moving symplastically, start from the outside, start inside the cell, will always move via the cytoplasm and the plasmodesmata. It will not have to change its trajectory when it gets to the Casparian strip because it never contacts the Casparian strip, just moves via plasmodesmata and eventually gets into the xylem. Transcellular movement, it enters the vacuole, which here is illustrated as the dotted line inside of the cell, and the vacuolar movement does move via plasmodesmata or apoplasts when necessary, but it ultimately, again, moves through the inside of the cell to get to the xylem on the inside of the root. So what about suberin and lignin make them not be able to conduct water anymore? Well, as evidence of hopefully how far you've come, I've taken this excerpt from Wikipedia about suberin in order to illustrate to you that you should be getting better at reading scientific texts. So let's see if we can make sense of this together. Suberin is a highly hydrophobic, meaning water-fearing, does not let water bind to it, and somewhat rubbery material. In roots, suberin is deposited in the radial and transverse cell walls. So that means the radial and transverse. It is put against the sides and the top and bottom, the sides and the top and bottom of the endodermal cells, the endodermis. This structure, known as the Casparian strip or the Casparian band, functions to prevent water and nutrients taken up by the root from entering the stele, or the xylem, through the apoplast. Instead, water must bypass the endodermis via the symplast. It has to move symplastically through the endodermis. This allows the plant to select the solutes that pass further into the plant. It thus forms an important barrier to harmful solutes. The endodermis can prevent things from getting in that shouldn't get into the plant, because after all, a plant doesn't have a choice in the water it drinks. I could pour acidic water on plant roots, and it would have to filter out the water and try to leave the acidic components in the soil. Plants don't have an option. They have to deal with what's around them. One of the adaptations that this Casparian strip enables in plants is, for example, in mangroves, these trees use suberin to minimize the amount of salt water they intake from their littoral or seawater habitat. So these are trees whose roots are able to pull water in despite the fact that they exist in salt water. Think about how crazy that is. If you were to drink salt water, you know that that dehydrates you. The Casparian strip in plant roots enables the mangrove to drink in salt water, but keep the salt out and move the water in. This is pretty cool, right? Now, in terms of some other details about suberin, we call it a lipophilic barrier, meaning it is a lipid-loving thing, and y'all know that lipids, like oils and water, do not mix, so a lipophilic barrier is something that will not allow water through. Suberin is a major component of cork. Like wine bottle corks. You'll notice that corks do a great job of keeping the liquid inside of a bottle to stay for long periods of time. I mean, corks capping off a bottle can keep the bottle from evaporating for hundreds of years, as is exemplified by, really old wine bottle finds. We additionally have suberin helping prevent water loss in the rind of melons. So if you've ever cut up a watermelon or a cantaloupe, you'll note that their outsides are incredibly shiny, or at least not conducting water, despite the fact that the insides are full of water, hence watermelon. Suberin is the polymer that's doing that, or chemical substance. And then we get suberin both in our primary, little bits of it, so thin cell wall, and our secondary cell wall, the thicker cell wall. Lignin is also another one of these polymers, an innovation that serves as a space filler alongside cellulose fibers. Now, lignin prevents the cell wall from absorbing all of the water. Because if the cell wall truly acted like a paper towel in that it could absorb all of the water, that would mean that it competes for water with the cells themselves. And we don't want that, right? You don't want water hanging out in the cell wall when it's needed inside of the cell proper. So to prevent water from staying in the cell wall, plants put lignin in between cellulose fibers, because just like suberin, lignin is again a kind of lipophilic barrier. It is great for water transport in the cell, or outside of the cell, because by having lignin in the cell walls, you are forcing things to move around apoplastically, because they're not just going to stick to the cell wall that they're attached to right now. Lignin helps scoot water along. Additionally, lignin has one more role of rigidity. Lignin is incredibly important in helping plants stand upright. It's a major component in woody cell walls. So we've got these two pieces, suberin, which is highly important in our Casparian strip, helping water leave the apoplast and filter through symplastically, and lignin, which is present in cell walls in large quantities, again, in the Casparian strip, as well as the xylem, helping our water move along apoplastically where necessary, but then also helping keep it out of the cell wall when the water needs to be filtered via symplastic transport. Now, not to intimidate you in any capacity, but to encourage those of you who are not intimidated by these molecules to maybe seek out a career in biochemistry. Because I think it's remarkable how large and complex suberin and lignin are. So I wanted to throw up these, and it almost feels literally like throwing these up. These diagrams of what suberin looks like. So you may have some primary cell wall here. This is actually kind of what lignin will look like. And then what suberin looks like in the lamellae, meaning the layered, the secondary cell wall. So you've got these things connected as these giant molecules that are really hard to break down. And you, as a human being, know this to be true. Lignin and suberin are difficult to break down. Do you eat the rind of a melon? Do you eat wood? You don't. These things don't get broken down very easily. This will play a huge role when we discuss uh, our breakdown of woods later, since we've discussed secondary growth already. It's this larger, more complex molecules that really make wood so difficult to degrade and ultimately led to fossil fuels in the first place. So it's thanks to the presence of largely lignin that we have any oil on this planet, because for a long time there was nothing on planet Earth that could break down lignin. And now I like this quote from my wife this morning when I was chatting with her before recording the lecture. She was a cell wall biochemist. That's what she did for a PhD. And I asked her casually, I was like, hey, do you remember how to draw lignin or suberin? And her response was, and I quote, well, they're waxy substances, so they're probably really large and messed up. So no, I don't remember how to draw them. And yeah, going to Wikipedia, she certainly was right. This here is lignin. And it took, and there are different versions of lignins, but this is a standard lignin molecule. You can see how much of this, or how complicated this extremely large molecule is, making it even more of a challenge to break down for planet Earth. But there are things that can break it down, namely some fungi. So we're actually going to end our lecture today by talking about fungi and their role in this entire pathway of water absorption. Because as we've talked about previously, yeah, sure, plant roots do an okay job of absorbing water. They have a certain amount of surface area that can absorb water and minerals from the environment. But I've already mentioned the fact that fungi can really enhance this process of water absorption through the formation of fungal roots, or mycorrhiza. And mycorrhiza is one of the terms that I misspell the most, so let's make sure we get it right. Mycorrhiza. Sometimes when you want to say plural, there's an E at the end of it. But myco is the prefix for fungi, and rhiza, suffix for root. So you've got fungal roots. And again, the whole exchange between mycorrhiza, which are a symbiotic relationship between a fungus and a plant, is that the plant produces a lot of sugars. It's got an excess of them, and it will trade the fungus, which does a great job of absorbing water and minerals and nutrients, because they're much thinner. Now, these fungi can be modeled interacting with our plant root as follows. You'll see the fact that this fungi is much thinner. Its hyphae, or fungal roots, are only a single cell thick and much smaller, meaning that their surface area is expanded compared to our plant. Compare the thickness of a hyphae to a root hair. Root hairs are, even though they're still one cell thick, they're much thicker. Therefore, their ratio of surface area to volume is less. Now, in terms of the plant root, you've got your epidermal layer, your skin layer on the outside, and your cortex. What you will see is that the fungus is actually able to grow into the plant cells and occupy a space with a structure called an arbuscule. So the fungus is now literally setting up a trading post inside of the cortex of the plant root. Now, additionally labeled, you'll see nuclei of the cells, the circles, and then you'll see some vesicles, some moving structures inside between the fungus and the plant. Now, it's important that these arbuscules are formed inside of the cortex, because looking back at our model of water movement, the plant ultimately wants to decide what minerals and nutrients and water get into the xylem or the stele. Therefore, the arbuscular mycorrhizal fungi, abbreviated as AMF, will set up shop in the cortex, so that way water can get dropped off here, and then the plant will do the last bit of filtering through the Casparian strip. These arbuscular mycorrhizal fungi never penetrate past the endodermis. They set up shop, set up a trading repository or trading depot in the cortex. The plant funnels down sugar into the cortex to give it to the fungus, and the fungus is absorbing so many of the minerals and nutrients and water that a plant needs. And so this is where we tie into our previous lecture. Well, most of this actually ties into the previous lecture in that they're all connected. This is all still connected to water movement inside the plant. But nutrition in plants and nutrient absorption is actually driven by the drinking in of water. So when a plant is absorbing water, it's also bringing in selectively through its plasma membrane specific minerals. In fact, plants themselves can be 10,000 times more concentrated in the minerals and nutrients that they need compared to the soil around them. In part, this incredible increase in concentration is driven by the fact that fungi are able to scour the landscape, scour so much soil, and bring those minerals into plant roots, and plant roots can selectively keep them inside while keeping out other harmful elements, like maybe lead. And so this relationship between fungi and plants, or mycorrhizal fungi in particular, in some cases can be connected to mushrooms that are edible. This is a member of the Boletaceae lesinum, so it's a cool edible mushroom found in North America and Europe. Member of the family Boletaceae, which is the porcini mushroom, which there are lots of varieties here. But this relationship is, this mushroom is one of the fungi that actually forms these mycorrhizal relationships with plants. So we eat some of the fruit from different plants, and then occasionally we'll also pick up the edible mushrooms, which are the fruiting bodies of these fungal networks that also help our plants get the water that they need. It is kind of cool to understand biology and dive further into the web of life. So with today's lecture, we've now discussed the structure of phloem, movement of water up the water column, and movement of water into the plant, and the filtration that is necessary. Next time, we'll wrap up our three-part series on water and internal plant movement by discussing the structure of xylem and the control of transpiration. See y'all next time.